So our next speaker is Keith Hamilton, coming from Rockefeller University from Regolutions Lab. And this was a switch in the program. We haven't gotten the most updated program. But we're going to go on to, after the introduction to tomography, sample preparation, next part of the workflow.
All right.
And Keith, what is your favorite part of the tomography workflow? What type of instrumentation?
The most frustrating and my favorite part is probably the fib milling.
This, okay.
It's the worst and the best.
Okay, this is the cautionary.
Because when it works, it's great, but when it doesn't work.
Okay, take it away, Keith.
So I'm going to talk about sample preparation. Basically, taking it from your sample, I'm going to focus on cells. You have cells, and how do you get them frozen? How do you get them onto an EM grid? What are the considerations you have to think about? And what are some pitfalls you might run into, and what are some things that you might be able to do with it? So through the talk, I'm going to go over the background and how you do things, and then present some test cases of things that I've done to show you things that can go wrong and things that can go right. So the goals, like I said, first one, get the cells on the grid. Second, freeze the samples. And third consideration is, how do you locate the target for milling or cryo-ET? So in the first part, getting the cells on the grid. So she talked about sometimes things don't need to be milled if they're thin enough already. So that's a consideration. Also, do the cells need to be positioned in the grid? So say some cells really like to go to grid bars, and they all just stick to the grid bars. So sometimes you have to get them away from the grid bars. And third, do the cells need to be patterned? Sometimes cells, say you want to look at stress fibers in a MEF or something, and you want the cells to be spread out. So how do you get the cell to adopt a specific shape in those grid bars? And then moving on to freeze the samples. Do the samples have the right geometry to plunge freeze? Plunge freezing is much easier than any other methods, such as high pressure freezing. And the thickness of the sample itself and the total mass are considerations you have to take into account. So, and also, do the samples need cryoprotectants? So cryoprotectants prevent non-vitreous formation. So again, things to think about. And locating targets during cryo, locating targets, you should probably think about this before you freeze. So you can label with fluorescent or gold, and we'll talk about that later. So preparing the grids for cell growth. You have an EM grid. If you grow the cells directly on the EM grid, the normal carbon support that you get on like a quantifoil or C-flat might not be sufficiently stiff or resilient for the cells. I've had cells that rip up the carbon, or cells that just aren't happy because the carbon is soft. But if you're just depositing cells onto it, you don't really have to worry about that as much. To say you have some bacteria you're just depositing, or some kilo cells, you're just going to stick them on. But if you are worrying about this, a lot of people have found that if you evaporate about 20-25 nanometers of carbon on top and bake it overnight at 60 degrees, it really adds a lot of resilience to the grid. And if you're fib milling, you don't have to worry about any thickness or that imparts. And another thing that you really have to think about is a lot of these culturing steps, there's a lot of picking the grid up, moving it to a different buffer, picking it up, washing it. And if you're going to pattern, and there's a lot of movement, and there's a lot of chances of bending the grid. Some people have really good hands and they can just kind of do this and they don't worry about it. But for me, I ran into a lot of issues. So people have published some stuff about 3D printed cases. This is from Florian Schur's lab. And you just print up this case and you put the grid in, and you handle the grid box, the case itself. You never touch the grid. I started using it. So there were some issues where the grid would float up when you put it in. The grid just kind of pop out of the case. And also, when I wanted to pattern on the grid bars. So I need to flip the grid over, pattern, flip it back over, and grow cells on it. So I developed in collaboration with, we have like a machining lab at Rockefeller. I developed these grids that it has a screw top, and you actually put the grid in there and you kind of screw the top on. And then you can both have it down the pattern and flip it up and grow the cells on top. And you never touch the grids from once you get them into the grid box until you're going to freeze them. And I made little things like a little thing, put it in 96 well so you can pick it up. This is the thing I use to pattern, so I put it in a little holder I glue down to a cover slip, and I primo pattern on that. And this is the case that I use to get it out. So I have a little, it positions it correctly to get tweezers in. I can unscrew it and take it out. So things like this, thinking about prior to doing your experiment, how am I going to handle the grid? Is the grid going to be resilient? These are really good things to think about before you even start the experiment, because once you're in it, you can waste a lot of grids and samples. So I talked about positioning the cells for milling. One thing that they have here and we have in our lab, and people have been using it more, is this thing called Primo, and it's a system that uses digital micro mirrors, like the projector right there. That you can project any arbitrary pattern onto through your objective lens onto your sample. So what you do is you put on anti-fouling coating, usually PLL PEG, or you can add in PEG SVA, and then on top of the PLL, polyL lysine. So that coats the whole grid, makes it passive. The cells shouldn't stick to it. I don't want a photo initiator, which is UV sensitive, and you let that sit, and then you put it on the Primo. The Primo illuminates the pattern and cuts away wherever the illumination is. So where the illumination is, there's no more PLL PEG. There's no PEG there, but there's still PEG wherever it didn't illuminate. And then you can go and put on whatever molecule you want, cell adhesive molecule, PLL, etc., and then put your cells on top, and the cells should stick to just where the pattern is. And you might need to play with the concentration of cells. It's something like sometimes you want to have it very dilute, so you have just one cell in the middle of the pattern and it expands, or maybe you just want to do it for a short period of time, so you have more cells. And this is just kind of on glass, a thing with MCF10A cells where I tried to get thin cell contacts, and I had just little dots with lines connecting it. And the fibronectin pattern isn't green, and you can see that it worked pretty well. So over time, the cells will deposit their own matrix and it'll go out of this, but it works decently well. And this allows you to get the cells in the middle of the grid bar, in the middle of the grid squares, and also allows you to get them in the shape that you want. So moving on to vitrification, the next part, getting it frozen. You need this to cool the entire sample down fast enough to prevent crystalline ice formation. This is it's not as simple as you might think. So you have to go from about 290 Kelvin down to 90 Kelvin in about 20 microseconds. So you have to do about 10 to the 6 degrees per second. And heat needs to transfer from your sample to, say, liquid ethane. And then from the liquid ethane away from your sample, so more heat can transfer out of your sample. So obviously, if there's more sample there, you have to transfer more heat out into the liquid ethane, and that has to transfer away. And all of the kinetics of that is very complicated. And sometimes some areas will be vitrified and some areas won't be vitrified. It's very you have to really test your sample, and this all limits this thickness of unfrozen samples to less than 1 micron. I would say much less than 1 micron. This is pushing it 1 micron. And the total mass in the grid. So even if you have a bunch of small cells, if you have a lot of small cells, there'll be a lot of heat in that liquid ethane surrounding it that you won't be able to kind of freeze, and you'll start to get non-vitreous ice. So, and also once ice starts forming, the heat of formation of the ice enters the equation, and you have to remove that as well. So it's kind of a runaway process. And one thing that happens is below 40 degrees, negative 40 degrees Celsius, homogeneous ice starts to form. So you don't need any nucleation point, and it forms very small ice crystals that don't really affect the overall structure. So if you look at a lot of old literature, a lot of it is freeze substituted. And they say, oh, you can freeze up to this size, you can freeze up to this size, and it's fine. But what it is if you remove the water and just look at the free substituted, all the structures and membranes look fine, but when you look at it with ET, it does not look fine. And I'll show you this. So this is this is a lamella that had incomplete vitrification. So you can see that these boxes are just where I collected tomograms, and this burning pattern there, that's indicative of incomplete vitrification. And when you look at the tilt series, as you tilt it, you see that it kind of blossoms at the different tilt angles, and that's indicative of the, as you're tilting, the crystal planes of the ice come into view. And these were kind of like microvilli type of things. And you can, interestingly enough, you can see that the crystal planes they're restricted to within membranes. So you can see that there's like a thing and it pops up. So it's almost like the ice crystal forms and hits the membrane, stops. And within these samples, especially this sample, I was able to get a low resolution ribosome structure. So you're able to get stuff, but the signal to noise is much, much lower. And it may distort the structure of embedded proteins. And like I said, you can counter with cryoprotectants, which would prevent the formation of crystalline ice, and also with high pressure freezing, which I'll talk about. So cryoprotective agents, there's two types of cryoprotectants: cell permeable and cell impermeable. Cell permeable, obviously it's going to be the best for preserving structure, but most commonly used is glycerol, and it can affect the cellular structure. So this is kind of a really old study that looked at yeast and adding in 9% glycerol after, and the cells shrink, and they see a lot of, like, the membrane is very distorted. They see some higher order structures arising and then they did some freeze fracture EM on this. And the yeast eventually recover, but it really is like you might distort a lot of structures if you add in the glycerol, even if it helps freezing. And people have frozen much larger cells using glycerol as a cryoprotecting plunge freezing. So if you're looking at just generally get a ribosome structure, maybe it doesn't matter. But if you're looking at, say, like I'm looking at actin networks, it really would matter if I added this in. The next is cell impermeable. So cell impermeable obviously just surrounds the cell, doesn't get in, which is good for preserving structures. But and you don't have to remove that heat of ice formation. But you're obviously not going to help the preservation within the cells or like prevention of ice crystals. And there's a study that came out from Blair and Scher again. They did some cryoliftat, looked at the extracellular matrix of cells, and they went through all these different things and found which ones had good vitrification and which ones also had less background. So if you're looking outside of the cells, these will just give a lot of background. Obviously, like I've had a lot of success with 20% BSA. So if I'm looking outside of the cell, it'll mostly be BSA and it'll just kind of be opaque. But if I'm looking inside of the cell, it's good. So these are also things you have to take into consideration, whether you're looking inside the cell, outside of the cell. So they looked at outside of the cell and they used 10% dextrin in phosphate buffer, and they found that gave a good vitrification without a lot of background. So just going on to actually freezing, liquid ethane unfreezing. So I use a one-sided blot, which is the GP. The VitrBot, you can also do one-sided blot. You basically just put on one piece of filter paper. So the filter paper goes up, touches the grid, and wicks away everything. I can replace this as 1/4 speed. You can see that there's a sample on top of the grid, and it moves through. It's not going anyway. It moves through and it doesn't wick it away. You have to wait a second and suddenly whoosh, wicks away. And then it moves more and more out and removes more liquid. And then you plunge freeze. And you have to remove, like I said, you need to cool the sample within 10 to 20 milliseconds to ensure that ice is vitreous. But this is kind of a paper that showed they just took a high-speed camera as they dipped a grid into liquid ethane. And it's very dynamic, the way that the grid first touches the ethane at one side, and then the ethane kind of goes up and then slams into the side. And during this whole time, there's cooling happening. So if something starts to cool by basically radiating heat. It's like the grid will act as a radiator. Up here, it'll start to cool slowly. And it might not be vitreous, but something down here might cool really quickly. And it's very dynamic. There's been a lot of, I've looked at, people have tried to figure out that I've worked with what part of the grid works better. If cells are touching the grid bars, is that better than cells not touching the grid bars? It really seems to be sample dependent. And you really, the proof is in the pudding. You have to look in the actual like lamella and see if it's actually vitrified. So this is just a test case, like I said. This is plunge freezing with no milling. And if the area of interest is thin enough, like I said, to be transparent, and will vitreous ice be vitreous after plunge freezing? Is it positioned in the center of the grid square? Blot time. So this was a sample that I blotted for 7 seconds. It's PTK2 cells. They're incredibly thin cells that they just happen to be really thin. They're not incredibly interesting cells, but we're just looking at kind of the actin networks at the edge of the cell. And you can see that this part, it's a very small part of the cell that you can maybe see that you can actually see through with the electron beam. And that part of the cell is so thin that it was vitrified. If you go in here, I'm sure this is all non-vitreous. But I was able to, again, collect tomograms. This was denoised with cryo care and segmented with dragonfly. So you can see that the blue is actin, green is microtubules, purple is membranes, and yellow is ribosomes. You're able to see really good structure. There was not really everything is vitreous. So this is the best case of you don't need to mill, and there's some small bacteria that you don't need to mill, but edges of cells. But most of the interesting stuff is not on the edge of the cells from very small bacteria. So this is a larger bacteria that was unpatterned. I just put it on the grid and plunge froze. And I did different plunge freezing times. And you can see that 1 second blotting, different blotting times. 1 second blotting time, there's still a lot of media surrounding the bacteria. 5 seconds, 10 seconds, it's mostly all removed. And no cryoprotectant. And when I looked at the SCM, the cells, you could see them as mounds. It's kind of easy to mill, incredibly easy to mill. And the lamella had really no sign. The cells were all vitreous. The only non-vitreous you can see here in this tilt series is outside of the cells where the media still was. There's no cryoprotectant there. A lot of the interior of the cell acted as a cryoprotectant for itself. So this is probably the edge of what can be used as just plunge frozen and still get no vitreous ice in the sample that you're looking at. So the next test case was, this was using a little bigger cells, and I patterned them in stripes to try and get them. I wanted to see them as resolved and away from each other as I could. So I kind of just I patterned on these stripes using the Primo system. I put on these EPH4, which are mouse memory cells, labeled with E-coherent fluorescent. And I plunge frozen on the GP with 15-second blot time. And you can see that this is what I showed before. There's non-vitreous ice, but I was still able to use it for some reconstructions, and I could see the structures. But it turned out that this wasn't physiologically relevant. It doesn't form good cell-cell junctions. The cells aren't happy. They want to form a sheet. So although I was able to get some good data out of this, it doesn't biologically make sense, which is like a lot. You have to think, you have to balance these sample prep, what's easy and doable with what's biologically relevant. And it's very difficult sometimes to balance these. So the next, I made donuts, and I made this donut because I found that if I patterned a circle of cells with a circle of cells not in the middle, it helped break the surface tension and drain a lot of the water. So I had very thin water on top. I also added on a little bit of detergent to get rid of any excess water, and I blotted for 15 seconds. And there was very little water on top, but I think the overall huge mass of cells on this grid, I don't think it was able to remove the heat fast enough, and the tomograms were barely usable. You can see that there is structure, you can see membranes and stuff in here, but it's not usable. So with the sample, I moved on to high pressure freezing. In high pressure freezing, the theory is you increase the pressure to about, it should be about 2000 bar, not 2000 millibar, 2000 bar before you freeze. So you increase the pressure and then you freeze. And in doing that, it slows the formation of crystalline ice, both because it increases the viscosity of the water and it also prevents the expansion needed to create crystalline ice. And this is kind of a phase diagram. It shows that when you go up to about 2000 bar, you go through ice, what's called ice 3 and ice 2, which is more of the amorphous ice, and goes straight down into this vitreous ice, avoiding the ice 1, the crystalline ice area completely. So you're able to cool down much slower, but by increasing the pressure. So in order to do this, you basically put your sample between two brass planchettes and put it in this little arm, close it up, put it in the machine. And this machine here, you put the sample here, there's a little lock thing you put it in, because if you don't, it would shoot up to the ceiling. It shoots in squeezed with high pressure liquid nitrogen, which is kind of cushioned by a layer of alcohol it puts in, because you want to pressurize before you freeze. So you pressurize with the alcohol that's being pushed by liquid nitrogen. The alcohol gets pushed out and then the liquid nitrogen touches it and cools really quickly, or decently quickly, much slower, about half as slow as you'd expect with a plunge freezing. So this might perturb cellular structures, but it's so fast it's probably not going to. There's things like actin depolymerizes at high pressures, but you're probably preserving it with this. It's within 20 milliseconds, it's probably preserved. So the first, I'll show you what this is the first way to do it. So you have this two brass planchettes, like I said, and you have a grid. And you concentrate your sample into like a slurry. And you just deposit the sample right into the upside down grid. And the upside down grid acts as little small wells. So you have the grid bars, and then between the grid bars, your sample kind of floats. And the carbon layer is on the bottom. So the gold grid bars are about 25 micron thick, so that's about how much you have. So you polish, the brass planchettes, there's a waffle method paper that goes up through all this. You have to polish them and coat them with hexadecine so they don't stick and you get nice flat surfaces. You high pressure freeze, and you can basically mill everywhere because the sample is just floating. The next case that was more applicable to me is high pressure freezing in a cured sample. So if you grow the cells directly on the grid, you add an aperture grid, which is just literally a grid of no grid bars. And you keep it from being squeezed. The different types of metals have different thicknesses. So you can kind of play with the thicknesses of the resultant sample. So you sandwich between the two brass planchettes that are polished and coated in hexadecine, high pressure freeze, and then you get basically a cell layer with a thin layer of the media layer that's dependent on the type of spacer used. And this is just what I did, the same type of cells. I used high pressure freezing, patterned a button to keep it in the middle away from so I didn't get that squished by the spacer. And the lamella showed the very little non-vitreous ice, and the tilt series had basically, there was very small areas of non-vitrification, but it was much better with the same samples than before with the plunge freezing. And this is just, I did another thing. I grew little cysts in there, and I got pretty good results. So you can do a lot of these things. You can grow different, like almost like organoid type things within the grid bars. There's a lot of different things that's just limited by your, imagination. But this type of setup. And so the last thing I'm actually going back. It's set up, but I'm in anytime. So labeling. Once you have the sample, you have it frozen, you have to be able to know where you're milling, unless the sample is everywhere. So you freeze yeast and it's just everywhere and it doesn't matter where you mill. You have to know where your target is. So say I'm looking at like some adhesion complexes. I need to know exactly where they are. Otherwise I'll be milling lamella and they'll be useless. So one thing about fluorescent tags is you can put a fluorescent tag in normally, like the normal, you can knock in a tag or just do like transient expression of a labeled protein. Leaching, really not an issue. I've just like imaged for ridiculous amounts of time on the same area and seen little bleaching. And membranes, especially multi-membrane vesicles, autofluoresce at cryo temperatures, strongest in the GFP channel, which most cells is not an issue, but I've run into some cells like MBCK cells had an issue where they secrete a lot, and there's a lot of like vesicles in there. Somebody I know was working on nerve cells and the myelin sheath was like ridiculous. It just, you couldn't see anything on the green, so we used red. But the thing is, you can't really localize. It's basically, you can kind of see where it is, but not, especially when you get down to targeting for like using tomography. And then after you get the tomograms, you can't really say, it's basically like the tomogram, there's a couple pixels that are from your CLEM. And so CLEM is correlative light and electron microscopy. So it helps to get you into the right area. But once you're looking at a tomogram, you can't say this is my protein and this isn't, because the signals basically over the whole tomogram. One thing that I've started to look at and other people have been doing is labeling the protein of interest with a small gold nanoparticle that allows precise localization in the tomogram. So this is an idea that came from old like pre-substitution type stuff, because you can just put in a nanoparticle label. It's not easy to get them inside of cells, obviously, because you want to keep the cells healthy and happy. That's why you're doing cryo. So you're limited in most cases to just extracellular targeting. And you also need to conjugate the gold nanoparticle to a molecule that will find your target. So other things that you can look inside cells are like gems, which some people have used. They're not as high contrast as the gold nanoparticles, obviously. And also ferrotags people have used, they're also not as high contrast, but they're available and you can think about using them for targeting. So, you know, if you're looking at a ribosome, you'll know where the ribosomes are. You can always find the ribosome, which is like it was talked about before. If you're looking for like a mitochondria, you can find the mitochondria, you can look for things on mitochondria, but a lot of times say you're looking for, I don't know, like a T cell receptor, like where is it? It's really hard to tell. There's a lot of things in the membrane, but if you label with the gold nanoparticle, then you'll be able to localize. And these are just some recent examples. I can't pronounce his name. So he has been adding fabs to gold nanoparticles, and this is sericilia, and he's able to look at the connection between the sericilia tips using this and localize and get a subtomogram average using localization. This is a recent one where they looked at synapses and they actually used a spy catcher tag and they expressed their protein with a spy tag on the protein, and you're able to kind of get in your need to use the fabs. And another recent one, which you can apparently get it into the cell, is this thing from Elizabeth Via's group, Exoslow Nano, where they take a bacterial toxin and basically put it in the membrane of the cell of interest, and then it pokes holes in it. You put your gold nanobeads in that are labeled with a halo tag, and it labels your proteins of interest, and then you wash the toxin out. So this is a new thing that I haven't really used at all, but you know, it's things like this. There is a lot of promise of people thinking of ways to get labeling inside of cells. And the idea sometimes is once you get enough data of labeled targets, you might be able to then train a neural network to pick unlabeled targets and then go from there. Because when you look at it, when you're trying to train a neural network on a cell, even if you have the structure, sometimes it's really hard to really get good training. So with that, I'd like to thank, so this is I'm in the elution lab, and all the people in the lab, I'd like to thank the CMRC, Mark's here, and Johanna, and especially Honkit has been really helpful with doing all the FID milling. The PIT, which printed all the cases, and I'll show you the cases during the practical, and the EMRC, which was helped me with the high pressure freezing. And I always like to think, even though my kid's not in the childcare at Rockefeller, I always like to thank it because without it, I wouldn't be able to have done anything. And the university has recently really given them a short shrift.
Thank you.
For the high pressure freezing, could you explain why liquid ethane is no longer needed?
Oh, so the reason you use liquid ethane rather than liquid nitrogen to cool things down is because liquid nitrogen will form a vapor as soon as it touches a warm surface. But in the high pressure, liquid nitrogen doesn't do that. And it's actually a much better cryogen and it cools things down quicker. But when it's high pressure, you don't get that vapor barrier.
Do you see like when you're blotting for longer times, do you see like a correlation between your ice thickness and whether or not your cells are vitreous? Like if you have a thicker layer of
Yes.
ice, is it better but harder to mill, or is there sort of like a trade-off there?
It's just not vitreous when you get a thicker layer of ice.
Okay,
Usually that's what, yeah. So the thinner layer of ice is basically just to get it vitrified. The milling, it's easier when there's no liquid, but you can do it. It's fine. It's really about you trying to get it vitrified, trying to get the less mass there to get the heat out from as you can.
so longer blotting times usually is more vitreous.
Yeah, usually, yeah, but then you also run into dehydrating this, like your sample. And
Yeah.
You might start, I've had a thing where I've definitely trying to balance. I image the cells before I freeze and then image the cells after I freeze and they look totally different. It's like, oh, it's probably, it was the blotting for too long. So, and then, yeah.
The form factor for the grid that we use for sample preparation right now has some issues like bars get in the way or the.
Yes, very much bars get in the way. So I generally, like I would use 200 mesh because that was the biggest that you use for single particle. Recently, I've moved to 100 mesh hexagons because I've found that I don't think, especially the high pressure freezing, you're not as worried about movement of the sample because it's a huge solid sample that you coat in platinum. So with the plunge frozen, yes, use 200 and we don't clip our own grids, the facility does, and I bother them to get them aligned with so you can there's like grid clips with notches. So I get them aligned because if the grid square is tilted, it's like a diamond, you basically can only mill a lamella right down the middle. If you go over to the sides, you'll hit a grid bar. So, but if it's straight on, you can basically run any lamella, but you still have to think about, is a grid bar in the way? Samples at the top near the grid bar, you can't do. Samples near the bottom of the grid bar, you can't do. You have to go right in the middle, which is another reason why getting the cell positioned or your target positioned right in the middle is so crucial for good milling. And I guess
So for the peg pure solution tree, how large is the peg chain?
the peg chain, I think 35.
Oh.
35, yeah, 3,500, yeah.
Do you have experience cryo-sectioning frozen samples?
No, I do not. But so I saw people do the lift out using, say, like the hydro to lift out. I did see a recent thing where they had like brain tissue and they cryo-sectioned and did actual like a microtome, and that's pretty interesting to me because you can make it thick enough that you avoid the squishing effects maybe of the microtome, but then you can then mill in more, and I think that's worth investigating.
For the gold nanoparticles, do they ever get in the way when you get to subatomic
Yes.
averaging?
Yes, because there's a huge delocalized signal from that. So
Right.
yes, that's why one of the things of using the fab rather than a smaller thing is you get a little further away and there will be that delocalized signal, which is one of the things of maybe if you can train a neural network to find it without that tag, it might be good.
Okay.
Any other questions? Yeah. Oh, wait, look, you.
Based on the different feeding method, which one is the best preferred for you?
So it depends. So if I just have a sample, the easiest is just plunge frozen because it's easy to mill, it's quick, but
treatments and a lot of the liquefaction, so like.
Yes, but if it's just, yes, that's the thing. So if I can do it and get it vitrified, I will love unfreezing. But other than that, I'll use the high pressure freezing. And there are other things. People are talking about the Vitrojet, which may or may not work to freeze things. So some people claim it can freeze very thick samples. You just basically shoot propane at both sides.
But that's very expensive, so it's not accessible to most of the users, right?
Yeah, no, and also I don't know if it really does work.
Okay, let's thank Heath